Hello everybody, I will be talking about a video of Richard Scientific. The human genome is the genome of Homo sapiens. That is the DNA sequence contained in the 23 pairs of chromosomes. In the nucleus of each diploid human cell, of the 23 pairs, 22 are autosomal chromosomes and one sex-determined pair, two X chromosomes in women and one X and one Y in males. If we look at the structure of genome, we can see, of course, a double helix structure. The code of life writing with only four biochemical letters, which we also call bases: adenine, cytokinin, guanine, and thymine. But how many are there in the human genome? Well, it's 3,000 millions. The DNA molecule is formed by the repetition of smaller chemical units called bases. There are four bases identified by the letters A, T, C, and G, by adenine, thymine, cytosine, and guanine, respectively. Two strands of DNA are paired to form the double helical structure discovered by Watson and Crick scientifics. The bases of each strand face or mate with the bases of the other, always following the same rule, versus A only T is located and only G is located opposite to C. It is site that two strains that are matured according to these rules and complementary. The complementary of bases allows the DNA to duplicate faithfully. For this first, the two strands are separated and each of them serves as a mold to manufacture. By means of enzymes of the cells, uh, another complementary and thus have two double helices identical to the original. That is, with the same sequence, the same information, once duplicated, and the information is transmitted equally from a strand cell to the outer cells through to mitosis or cell division. Another type of cell division, called meiosis, occurs in the mother cells of the gametes, sperm in man and eggs in woman. In meiosis, the number of chromosomes is halved. The DNA the DNA sequence that makes up the human genome contained the encoded information necessary for the expression highly coordinated and adaptably to the environment of the human proteome. That is, the set of proteins of the human begin proteins and not DNA uh, are the main effector biomolecules. They have structural, enzymic, metabolic, regulatory, signaling functions or organizing the cells in hook, functional networks or interaction. In short, the proteome bases the particular morphology and functionality of each cell. Likewise, in the structural and functional organization of the different cells forms each tissue and each organ, and finally the living organism as a whole. Thus, the genome contains the basic information necessary for the physical development of a complete human being. In a study carried out by certain well-known scientists after 15 years with approximate budget of $4 billion, the published the genome sequence. The possibility of universally diagnostic using the genome is possible today. Today, it is possible. The importance of this for all of us could live 5, 10, or 20 more years of light, all thanks to the genome. It is an excellent new unless we consider our ecological footprint on the planet and our ability to maintain food production, but it turns out the same technology is being used to create new varieties of corn, wheat, soybeans, and other crops, which be much more resistant to drought, floods, pests, and pesticides. Now, as the world population continues to increase, we will have to continue development and eating genetically modified food. The alteration of DNA sequences that constitute the human genome can case the abnormal expression of one or more genes, uh, originating a pathological phenotypic genetic disease can be caused by the mutation of DNA sequences by affecting the coding sequences, producing correct proteins, or by regulation sequences altering the level of expression or of a gene, or by chromosomal numerical structural alterations. The alteration of the human of the term self and individuals is frequently determined to the discipline. Currently, the number of known genetic diseases is approximately 4,000. The most common being cystic fibrosis. The study of disease has often been encompassed within the genetic of population. The result of the human genome project are of great importance for the identification of new genetic disease and for the development of new and later system of genetic diagnostics, as well as for research in new treatment, including gene therapy. The scientific Charles Resnick asserts that all these disorders can be solved analyzing the sequence of the chromosome. In this way, a correct a correct correction can be made to the patient whatever he thinks that with the technology of his company, it is possible to achieve simultaneously compare a chromosome of two or more individuals with the objective of knowing their compatibility, even knowing if they share the same blood, in the sense that even they are family. In my opinion, this scientific is is very amazing that in his video uh, he talk about information very important of DNA and the technology that he use in your company. Thank you.